Hello, this is Dr. Pflügel, your instructor for the laboratory course LS23L. If you have any questions, please contact me at drpflügel@gmail.com. The following presentation will cover some background needed for your next laboratory. In this week's lab, you will be working with E. coli cultures, use an enzyme activity assay, and we will introduce the use of the spectrophotometer. The concept covered in this lab will include understanding of the lag operon and its function, using a protein activity assay to determine enzymatic activity, and using the spectrophotometer to determine cell density. The laboratory skills you will meet in this lab include the proper use of micropipetters, correct handling and disposal of bacterial cells, using the spectrophotometer, including a correct blank. This lab will also continue to enforce standard lab safety. So remember to bring your lab coat and proper lab attire as explained in the lab safety video. Otherwise you will miss your pre-lab quiz or even a larger part of the lab. In the following lecture, I will present some background on the lag operon, enzyme activity assays, and measuring cell densities in liquid cell cultures. You will learn to measure the activity of the enzyme by measuring the rate at which product appears and the time required for the enzyme to be synthesized. You should have some background on this from LS3. However, the following lecture will go over the basic concepts of the lag operon and laboratory techniques used in the enzyme activity assays to refresh your memory and to prepare you for the actual lab. Since you will be completing the worksheet in your lab, it will help you to understand the content of the lab much better, if you know the background beforehand. The protagonist of this week's lab is lactase. Lactase is an enzyme that breaks down lactose, which is a disaccharide, into two monosaccharides, galactose and glucose. Since it hydrolyzes a beta-1,4-glycosidic bond, it belongs to a class of proteins referred to as beta-galactosidases. Lactose is a sugar found in milk and all mammals will express lactase at birth. However, most mammal species will not consume milk after infancy and so do not express lactase as an adult, which means they are lactose intolerant. All dairy products do have lactose and are easily digested for people from European origin. However, the ability to continue drinking milk as an adult is a result of a mutation. The mutated gene never sends a signal to turn off lactase production at about the age of weaning. If you don't have that mutated gene, then you are lactose intolerant, which can be very uncomfortable, since if you don't digest the lactose in the milk, your bacteria will do it for you, and they will produce a lot of gas as a byproduct. Humans have probably always had occasionally this mutation, but it didn't start to spread through the population until humans started using dairy in their diet, a known 8,000 years ago, and probably earlier than that. However, the distribution of this mutated gene is not as global as one would think. The map shown here shows the percentage of the world population that is lactose intolerant, which means have difficulty digesting dairy products. Huge amounts of investigation of DNA have more precisely traced those pathways, and the spread of the lactose tolerance gene is one of the best studied subjects in modern genetics. Lactase and its regulation has been extensively studied in E. coli. E. coli is a common bacteria used in the laboratory and uses a series of genes, the lag operon, to metabolize lactose. E. coli has one chromosome and several plasmids as shown here on the top right. The single chromosome is actually circular and it contains two regions which includes the lag operon and the lag repressor. Before we go into any more details, let's review gene expression in general. Here is a very short review of the process of gene expression. or how a gene on a segment of DNA is transcribed into an RNA molecule, and how that RNA is then translated into a protein, which carry out the vast majority of the functions of a cell. The first step in gene expression, which is the process of transcribing a DNA sequence into RNA, is called transcription. Most genes contain a region called a promoter that lies about 10 bases before the beginning of the gene. This region marks the point where the enzyme RNA polymerase begins transcription. Without this promoter, the RNA polymerase will not recognize this region of the DNA as containing a gene, and transcription will not occur. Following transcription, the mRNA is recognized by the ribosome, a large complex of proteins and RNA that is the site of protein synthesis. This process of translating a sequence of RNA into a sequence of amino acids is called translation. The activity of a specific enzyme can be regulated by inhibiting the protein directly or by regulating its transcription or translation. Lactase is regulated on the transcription level with a series of genes which are called an operon. An operon is a series of genes whose expression is coordinated together. This lab focuses on the lag operon, which is the first and best studied example of gene regulation. At its core, the lag operon is a mechanism for E. coli to detect the nutrients in its environment and to do different things in each case. The lag operon includes 3 structural genes that are all involved in lactose metabolism. The 3 genes are called LAG-Z, LAG-Y, and LAG-A. The operon also includes the regulatory elements that control the expression of these genes. The 3 genes make up a unit that shares a common promoter, also known as LAG-P. In addition, the promoter, there is also a region called the operator, also known as LAG-O. Only the 3 later genes actually code for proteins, which are shown here, and therefore these genes are also called structural genes, as to differentiate them from regulatory elements, such as the promoter and operator. Shown here is the lag operon and its location on the chromosome. The operator is the binding site for a protein called the lag repressor, which is a key for the regulation of the lag operon. The lag repressor is encoded from a separate gene called LAG-I, which is located elsewhere in the genome. It has its own promoter and is not part of the lag operon itself. Here are the locations of the enzymes encoded by the lag operon in the cell. LAG-Z encodes lactase, which will be found in the cytosol. LAG-Y encodes a channel protease called permease, which will be found in the plasma membrane. Finally, LAG-A encodes a minor enzyme that chemically modifies lactose during its metabolism, which is also located in the cytoplasm of the cell. The lag repressor gene product of the LAG-I gene will also be found in the cytosol. Now, let's have a look at each of these proteins which are involved in the lactase regulation in detail. LAG-Z encodes lactase, which actually performs 2 functions. Primarily, it breaks down lactose into galactose and glucose, both of which can be used for energy. However, a minor function of lactase is to isomerize lactose into a molecule called allolactose. Allolactose is an isomer of lactose, differing in its sugar-sugar linkage. For our purposes, allolactose functions as an indicator of the presence of lactose in the cell. I come back on the importance of allolactose later on. For simplicity, I will use following symbols for each of these molecules in the following introduction to each of the proteins involved. LAG-Y encodes a channel protease called permease that allows lactose from the environment to enter the bacterium. Without this channel, lactose is unable to enter the cell. Remember that sugars tend to be polar molecules that can't cross the lipid bilayer. Here on the right is a figure with a small overview, with two of the three enzymes coded by the LAC operon. Finally, LAC-A encodes a minor enzyme that chemically modifies lactose during its metabolism. This enzyme is not important for our purposes, and we will not go into details here. The LAC-I gene codes for a protein, the LAC repressor, shown here on the left, which is a DNA binding protein. The LAC repressor protein contains two functional domains. One domain binds to the operator region of the LAC operon, creating a DNA loop, and the other domain binds allolactose, which is formed in bacteria in the presence of lactose, as mentioned earlier. When the repressor binds allolactose, it changes its conformation and dissociates from the DNA. Just note, this detail is different in the lab manual. In the lab manual, lactose is listed as the molecule which binds to the repressor. However, it is actually allolactose which binds to the repressor. This slide shows all the components of the beta-galactosidase regulation. Here on the right is the LAC operon, and here on the left the genes for the LAC repressor, which has its own promoter. RNA polymerase will bind to the promoter region and transcribe the gene. The repressor gene, mRNA, is then translated by ribosomes into the repressor protein. The repressor binds to the operator region of the LAC operon, creating a DNA loop involving the promoter for the three structural genes, and thereby blocking the translation of these structural genes. Normally, in the absence of lactose in the environment, there is a minimal level of allolactose in the cell, and the LAC repressor stays bound to the operator. This acts as a physical obstacle to the binding of RNA polymerase to the nearby promoter, and as a result, the three genes, LAC-C, LAC-Y, and LAC-A, are not transcribed into RNA, meaning that their corresponding proteins aren't translated either. However, in the presence of lactose, some of it is converted into allolactose. And when the allolactose binds to the LAC repressor, it induces a conformational change in the repressor that eliminates its ability to bind the operator. As a result, the repressor is no longer able to block the binding of RNA polymerase to the promoter. The three LAC genes are then transcribed into mRNA, and the three proteins encoded by the genes are synthesized. Once the LAC genes are expressed, lactose is metabolized to galactose and glucose. Since allolactose is a molecule which switches on the expression of the structural genes, it is called an inducer of the LAC operon, and the process of switching a gene on is called induction. Notice also that this forms a feedback system as well. As lactose is broken down during its metabolism, the level of allolactose drops as well. After a certain point, there is no longer enough allolactose to bind to the LAC repressor, and the repressor reverts to the conformation that remains bound to the operator. In effect, the expression of the three genes will stop, and the lactose in the environment is used up. A very efficient system indeed. Here's a summary of what happens in the presence and absence of lactose. Pay attention to the net result of having lactose in the environment. In effect, the three genes involved in lactose metabolism are repressed when lactose is absent. This is important because it is wasteful for the bacterium to synthesize machinery for a nutrient that is not actually present in the environment. However, as shown on the left, the LAC repressor is always expressed and present in the cell. As we have seen earlier, when lactose is absent, the LAC operon is repressed. What might be confusing is the transition from no lactose in the media to lactose in the media. Without lactose, there is no lactase and no permease. So if we add lactose to the media, how does it get into the cell? And even if lactose finds its way into the cell somehow, how does lactose get converted into allolactose if there's no lactase present? This can be explained by the fact that the repression is not absolute. The binding of the repressor is dynamic, and occasionally the repressor will detach and RNA polymerase might be able to bind and transcribe the genes before another repressor binds to the operator. In this way, an extremely small amount of proteins coded by the 3 structural genes can be made even when the operon is repressed. Here's an updated summary. Without lactose, the structural genes of the operon are expressed at extremely low levels, however, generating less than 10 molecules of beta-galactosidase per cell. When glucose is absent and lactose is present, the lac operon expression is enhanced at a level that is 100-fold higher than without induction. The mechanism is slightly even more complicated, however. Many bacteria, including E. coli, prefer glucose as a source of ATP because, for example, lactose is a more complex molecule and its metabolism requires the synthesis of special proteins like beta-galactosidase and is therefore energetically wasteful. Essentially, if glucose is available, bacteria utilize it first before resorting to other energy sources. The 10 genes for the glycolysis pathway, which are shown here, are always expressed and present in the cell. When E. coli detects the presence of glucose, it executes a decision using a protein called catabolite activator protein, or CAP. CAP, not unlike the lac repressor, is a DNA binding protein and has 2 states. If CAP is binding a small molecule of cyclic AMP or CAMP as shown here, CAP will change its conformation, which allows CAP to bind to an activator region near the promoter. If there is no CAMP, then CAP doesn't bind to the activator region. The CAP protein interacts with a region near the lac operon promoter called the activator. This interaction helps RNA polymerase find the promoter and in effect increases the expression of the 3 lac genes. Whether or not CAMP is present in the cell to bind to CAP, in turn, depends on glucose levels in the cell. Here's an overview of the lac operon with and without glucose present in the presence of lactose. When glucose is low in the cell, CAMP is high, and CAP is able to bind to the activator. As a result, expressions of the lac operon is enhanced, and the cell utilizes lactose as its energy source. When glucose is high in the cell, CAMP is low, and CAP is unable to bind to the activator. As a result, expression of the lac operon is kept at a low basal level, and the cell uses glucose as its energy source. Now, let's move on to the actual lab. How is the expression of lactose metabolizing genes actually studied? Shown here on the left might be a representation of a cell without induction. Lactase is at a basal level. On the right, we are looking at an induced cell with 100 times the amount of lactase. In the lab, we will primarily use lactase activity as a readout or representation of the lac gene expression, so we will be measuring the enzyme concentration indirectly by its activity. The enzyme activity assay itself is actually done outside the cell in vitro. In order to take exact time points, we have to break open the cells to get to the enzymes. Now, to obtain active enzyme, we want to stimulate the production of lactase, and one way to increase the expression of the lac genes is to add lactose to the bacteria in the absence of glucose. However, there is a problem. Remember the feedback mechanism? While adding lactose will increase lactine expression, the proteins produced as a result will reduce lactose levels over time. Without enough lactose in the cell, there won't be enough allolactose to prevent the LAC repressor from blocking gene expression by binding to the operator. This means that LAC gene expression will die out over time if lactose is used to stimulate the expression, which limits our ability to study the LAC operon. To get around this, there is a substance called IPTG, which is a molecular analog of allolactose, meaning that it is a synthetic molecule structurally similar to allolactose. However, unlike allolactose, or lactose for that matter, IPTG cannot be broken down over time, meaning that the expression of the LAC genes is maintained at a high level. Therefore, IPTG is known as an inducer because it induces high expression of the 3 LAC genes. Importantly, IPTG cannot be broken down by beta-galactosidase, so it theoretically stays at a constant level in the cell. On the other end, we also want to have a way to measure the activity of beta-galactosidase, and the end products, galactose and glucose, are not easy to detect. To do this, there is a substance called ONPG, which is an artificial substrate for beta-galactosidase. It is similar in structure to lactose. Lactase hydrolyzes ONPG into galactose and a product called orthonitrophenol. ONPG is colorless, but orthonitrophenol happens to appear yellow in color and absorbs light around 420 nanometer, which is the complement of the color yellow as shown here. In lab, you will use spectroscopy to measure the amount of orthonitrophenol in your samples, which in turn will give you a quantitative measure of the level of beta-galactosidase activity. Notice that together, IPTG and ONPG separate the 2 functions of lactose. 1, to induce expression of the LAC operon genes, and 2, to act as a substrate. The enzyme activity assay itself is actually done with ONPG outside the cell in vitro. For that, we have to break open the cells and extract the proteins. So now we have a system to determine the expression of lactase by its activity. However, there is a time factor. To switch on the operon and to express the genes takes time. Furthermore, while we wait for any amount of time, the E. coli cells in culture will keep growing too. To correctly estimate the amount of enzymes produced per cell, we have to take into account the number of cells in our test tube. Over time, the cells will multiply. For instance, the bacterial growth rate is a function of resources, and so the uninduced cells might grow slower than the induced cells. How can we compare gene expression at different time points or in between 2 different experiments, for instance, induced cells versus non-induced cells? The answer is that we basically need to normalize the total enzyme activity of the sample by estimating cell density. Shown here are 2 glass tubes with bacteria. The left tube shows LB media for a bacteria culture, and on the right, the same media after several hours of incubation. It looks turbid, which is an indicator of bacterial growth. So how can we estimate the number of bacteria in a sample such as this? One method is by using spectroscopy, measuring the optical density at 600 nanometers. In case of live bacterial culture, we don't measure the absorption of a wavelength, but the amount of light scattered by the bacteria in the sample. Therefore, in our lab, we will be using 2 measures. The OD420 reading for the activity assay, and the OD600 reading for the live bacteria. This might confuse you, so make sure you understand the difference when you are performing your experiment next week. For the enzyme assay itself, Time is also important. Since the longer the enzymes are active in solution, the more product they will produce, until the educt is depleted. Therefore, to compare different samples, we have to stop the reaction at specific times and then normalize the activity by time. In the actual experiment, you will break the cells open with chloroform to stop the gene expression at an exact time point, and you will stop the enzyme reaction in the activity assay by changing the pH. Since there are several time points you need to keep track of during the experiment, make sure not to confuse the bacterial incubation time with the time used in the activity assay, which is a common mistake students can make.